In this video, you'll learn about bone formation through the processes of intramembranous and endochondral ossification. Ossification is the process of bone formation by replacing another tissue with osseous tissue. Intramembranous ossification is where a bone develops within a fibrous membrane. For example, within the dermis of the skin, we have bones forming. Examples of major bones that form through this process of intramembranous ossification include the parietal bones, the frontal bone, the clavicle, and the patella. Intramembranous ossification starts with the differentiation of osteoblasts. These osteoblasts form within the embryonic connective tissue known as mesenchyme. This structure, consisting of osteoblasts that are starting to secrete osteoid, is known as an ossification center. As the osteoid continues to grow, surrounding osteoblasts, they mature into osteocytes. And the connections of osseous tissue are known as bony spicules. Bony spicules continue growing until they connect surrounding blood vessels. So blood vessels become entrapped in this meshwork-like structure of osseous tissue. Now that the bony spicules have grown together, they've formed what are known as trabeculae, and the blood vessels are within the spaces, the trabecular cavity. So this is essentially the formation of spongy bone at this point. Next, the superficial layers of the bone will start to form into compact bone. Osteoblasts organize around the surface into the newly formed periosteum and start to ossify the superficial surface, forming compact bone. Endochondral ossification is the formation of bones from a hyaline cartilage model. Here we can see the formation of a long bone from a hyaline cartilage model, where the outer membrane surrounding the cartilage is perichondrium. As the cartilage model grows, the cells that are deep inside start to become starved of nutrients, and the matrix surrounding them becomes calcified. Then those chondrocytes die, leaving space. Blood vessels start to grow inside of there, and they carry in with them osteogenic cells that differentiate to form osteoblasts. The osteoblasts start to secrete osteoid and create a primary ossification center. The superficial membrane surrounding this structure now differentiates into a periosteum. So the periosteum has osteogenic cells that can mature into osteoblasts on the superficial surface, and the blood vessel carried in osteogenic cells that are differentiating into osteoblasts to create this primary ossification center. Osteoclast cells also migrate in with the blood. Within the developing diaphysis of the long bone, osteoclasts start to break down some of the bone in the center of this primary ossification center, creating the medullary cavity. This process that created the primary ossification center repeats within the developing epiphysis. So we have chondrocytes that start to become calcified and trapped within this calcified matrix. They enlarge and die, leaving space that blood vessels grow into, carrying osteogenic cells in. Those osteogenic cells differentiate to form osteoblasts that secrete osteoid, in the secondary ossification centers. This occurs at both of the developing epiphyses within a long bone. Last, the periosteum has these osteogenic cells that can form a superficial layer of compact bone as the osteoblasts start to secrete osteoid on the surface. Compact bone gets laid down superficial to the spongy bone that was formed deep within. Now we have the mature structure of a long bone, the structure that is present at birth, where there's still hyaline cartilage present at the epiphyseal plate, and at the end it has the articular cartilage to cover the joint surface.